Just a reminder that Lynn has offered to lead a review session that will meet here. Lynn, where are you? Yeah, you'll meet here after class. So in other words, a little bit of a marathon there, probably at 4:30, right? So they can go and grab a quick something and come back in. Okay. Any other questions before we proceed? Is that sound reaching fairly well? It's hard for me to judge. Okay? All right. All right, then let's plunge ahead. So, come on. Oh, that's 2007. All right. Okay, I'll do this then. What's the deal here? Okay, there we go. Oh, please. Try it from here then. There may be some small differences in this file than the one we had over there, but I think it should be close enough. So, if we could have a little more quiet, we can probably get going on this. Thank you all for finding this room. I apologize for the moves around. It's not of my choosing, you can imagine. But I appreciate it that so many of you have, in fact, found it. So as I believe we did discuss termination then, is that correct? Okay. The process of termination in a prokaryote is related, but not identical. In prokaryotes, there is a ribosome recycling factor. Looks something like that. And it cooperates with EFG, the elongation factor G. But again, you can see this thing that kind of looks like a transfer RNA faking out the ribosome, so to speak. So the ribosome release factor can bind at the aminoacyl site, the aminoacyl site. EFG will move it over, and that will move the deacylated tRNA out altogether. After the hydrolysis, the ribosome will then fall apart. And once the ribosome falls apart, the messenger RNA is released and can go on for another round of initiation yet again. And that's obviously out of date. Reminder that the final exam will be May 13th, Tuesday, and it's at noon, if I remember correctly. And we will announce the rooms, but we will be divided between two rooms. And maybe this is as good a time as any to say this. Because we're divided between two rooms, I will send an email that specifies which of the two rooms I would like you to come to. We don't have enough space for everybody to pile into one. And so for the final exam, unlike the others, we'll actually ask you to show some form of ID with your name on it, so we know that you're coming into the right room, so we don't end up with too few tests in a particular room. Do people have other questions about the final exam before we go on? It is cumulative. There will be a week of review coming next week that should be very helpful to you, I hope. There will be a number of questions on the final from this year's hour exams. So using those as a review tool should be helpful too. And there will be no essay bonus question. I'm just kind of curious. The question, in case people didn't hear it, was, can I tell you what percentage of the previous exams will be on the final? I really don't see what useful information that conveys. And for another thing, I haven't written the test yet. So in that sense, it's still a matter of the future. All right. We've been talking so far as if a ribosome and a messenger RNA were a one-on-one relationship, that for each message there's one ribosome sitting on it and that's all that's going on. That would be misleading. That would be misleading. It is far more common to have more than one ribosome reading a message at once. There has to be a certain amount of space between the ribosomes for there to be room for them to move. But a messenger RNA is a large thing. And it's not uncommon at all for one messenger RNA to have 3 to 6 ribosomes on it. Large messages like myosin heavy chain can have 15 to 18 ribosomes on them and get very big indeed. When we look at the structure of eukaryotic ribosomes, there's the 5' methyl cap at the 5' end. There is a cap binding protein that is among the initiation factors. There's a 3' tail that was added post-transcriptionally by an enzyme that just adds A's, no template involved. And the poly A binding protein that recognizes poly A. These two interact and bind to each other so that a typical eukaryotic ribosome is actually a ring-shaped structure. And one of the things that buys for eukaryotes is it confines initiation to messenger RNAs that are complete. That without this kind of a provision, you might waste a significant fraction of your effort trying to translate things that in fact are only still half there. This picture underneath it is an electron micrograph, meaning a microscope with magnetic lenses and electron beam. And you can actually see individual ribosomes on this. So the strand that's running through the middle is the messenger RNA. The larger blobby things are the ribosomes themselves. And then the smaller kind of branches coming off are growing protein polypeptides. This must be a fairly large protein, looking at the length of this polyribosome. Well, this trick with the 5' cap binding protein and the 3' poly A binding protein works very well for eukaryotes who have 5' methyl caps and poly A tails. Prokaryotes don't have those things, and so how do they make sure they don't waste their time on bad messages? And there's a neat sort of an insidious trick that the prokaryotes have instead. They have a special thing called ssRA RNA, which is a small, stable RNA, about 10S, and encodes 10 amino acid residues. So part of it is a mini coding sequence. Other parts of the sequence fold up into something like a tRNA. And that tRNA can be charged with an alanine. And so this is a kind of a funny RNA. It's both functioning sort of like a messenger RNA and sort of like a tRNA. And so what it looks like is something like this. Here you have a ribosome that's got a peptide chain sitting in the P site. And this chain can be transferred to the alanine, and okay, let me see how I can express this. When it's so if you have something that has gotten stuck on the ribosome and it's not properly released by the release factor. Remember we talked about the RRF, the ribosome release factor. The ssRNA can bind in that empty site that's not releasing properly. The growing peptide chain will be transferred to that little alanine. And the messenger RNA that was defective in that sense is released. And that RNA can now be degraded. That won't get reused. That's an aspect of messenger RNA quality control. But we still have, sitting on the ribosome itself, the ssRNA with the growing chain that was transferred to it. It slyly moves over, and its little mini open reading frame, which codes for 10 amino acids, is used to translate those 10 amino acids. And when it's completed at this point, then the protein that is released in this way has this tag on it. And that tag is one that says, this came from a bad messenger RNA, degrade me. And so that's an aspect of protein quality control. So it's a very different method for quality control than the eukaryotes have. And this is showing you how the ssRNA works in a little higher magnification, if you like. That you have the stalled messenger RNA here that can't terminate properly. It can't terminate properly because it doesn't have a stop codon, got damaged somehow. So you have a stalled ribosome like that. ssRNA can come in and sit at the A site, properly accept the transfer of the peptide chain through the transpeptidase. When it has done that, the ssRNA moves over into the P site, and the old RNA is released and degraded. And this little bit of coding sequence swings around and slides along in there so that you have most of the ssRNA out of the way, just its little coding bit working. And there's that tag on that peptide that marks it as time to get rid of this. And it does have a proper stop codon, and that's where the release factor recognizes the stop codon and opens up the ribosome for future use. Translation is not enough. Just to be turned into a linear polypeptide will not by itself complete everything that has to happen before a protein is going to be useful. At the very least, proteins need to fold. Some proteins are processed in one way or another. Some proteins have to translocate. They have to move from one compartment to another process. And this is just a little schematic of protein folding. We talked about protein folding a little bit earlier, back when we were in the structural part of the course. And if we have this little polypeptide chain like this, with the red bits being the hydrophobic parts, if this thing folds up in a proper way, it makes this compact structure with its hydrophobic bits in the middle and the hydrophilic parts on the outside. On the other hand, it can misfold. It might misfold into something like this, which is not very stuck. It might then unfold again and refold properly. Or a number of them might end up together in a tangle where several peptide chains together have sort of lumped all of their hydrophobic stuff into the middle. This kind of a tangle is the sort of thing that's seen, for instance, in Alzheimer's disease, where proteins form amyloid plaques. And we used to think that protein folding was something that just happened spontaneously. And we've discovered in reasonably recent years that although some proteins indeed can fold well by themselves in dilute solutions, in the cell, there are other proteins around that help with the process called chaperones. Now, the word chaperone may be sort of a blast from the past. I don't know if any of you have been in situations that actually had chaperones. But in the old days, the role of the chaperone was to sort of sit around and to make sure that there weren't any interactions happening that shouldn't happen until the right time. And that's more or less the job of the protein chaperones. And one of the reasons that this turns out to be important is that the interior of the cell is far more crowded than the typical test tube that we do our experiments in. So the cytoplasm can be as much as 30% protein by weight. And it's a very heterogeneous mixture of proteins, so that as a growing peptide chain comes out, there's no guarantee at all that it will have a free space in which to fold up properly. This is an example of one of the multimeric chaperones. We're going to look at that, I think, in just a little more detail in a second or two. Yes, we will. And so you can see that this one is really a fairly substantial structure that's committed to the process of protein folding. Another group of proteins that helps with protein folding, or which can keep these hydrophobic bits from mushing together to make a mess out of things, are the heat shock proteins. They were called heat shock proteins because they were first discovered as proteins that become more abundant if you expose cells to higher temperatures than they like, heat shock them. But heat shock proteins are also evoked by many different stresses to the cell, And it turns out that one of the reasons that they're evoked by many different stresses is that anything that can cause denaturing and misfolding is a challenge to the cell, and the heat shock proteins can help with that. They tend to bind to the hydrophobic domains of things, and then will release it when they hydrolyze their ATP. If things don't go right, they may come back and try again. This is a picture of how one particular chaperonin works. Here you have sort of the basic state of it, if you like. And we've got our stray wandering fellow over here that we want to help fold correctly. For it to enter this chamber, actually has to bind ATP before it's functional. And the hydrophobic bits of this peptide will be interacting with hydrophobic bits in the, on the GRO-EL. Then we have a GRO-ES protein coming in. It closes off the top of that chamber there, and the in the process there's fewer hydrophobic patches involved, exposed for things to interact with, so that the peptide that's stuck in there has itself to interact with, basically. The GRO-EL will hydrolyze its ATP, and in the process, it'll have a chance to release the peptide when the next round of ATP and peptide come in at the opposite end. So it's kind of a one end does it, then the other end does it, and so forth. So when this chamber gets filled up, it releases the top chamber, and then it can go through another round. This means that for each folding event, the cell is investing about 7 ATP. Well, considering how many ATPs it costs to do protein synthesis, this is a fairly small investment in quality control. And it's not every protein that need it. Maybe 15 to 20% of the proteins need the assistance of chaperonins in order to fold properly. There are a couple of other kinds of help that proteins have in rearranging. One is called proline isomerase. You may remember the structure of proline, which is the amino acid that has kind of a ring structure to it because it folds back on itself. And that structure is not very readily rotated on its own. What proline isomerase will do is assist a protein to switch a proline from one state to the opposite state so that it can explore the full spectrum and hopefully find the right final folding state. Protein disulfide isomerase, you may remember that cysteine has that sulfhydryl side chain and that some proteins make disulfide bonds, sulfur to sulfur bonds. If you have more than 2 cysteines in a protein, you might make that to the wrong cysteine. And what protein disulfide isomerase does is let proteins explore other cysteines to try to come up with the correct solution. Not everything that is made needs to be in the bulk cytoplasm. Some things need to be in other parts of the cell. Some things need to be outside the cell. And so some proteins have to go into or through, completely through, membranes. In a prokaryote, it is the completed protein that is transported. That is, translation happens, and then the cell makes the decision this should be somewhere else. Interestingly, in the organelles, it is also only after the peptide is complete. In eukaryotes, there is a method for co-translationally inserting proteins into and through membranes for the ER, for the single membrane-bound components of the cytoplasm, and for things that need to be exported completely outside the cell. So if a protein is going to go into a chloroplast, or if a protein is going to go into a mitochondrion, that transport is principally with completed polypeptides. But if it's going into a lysosome, or if it's going into a glyoxysome, or things like that, that would take advantage of the pathway, the co-translational pathway. And the way the cell has for going about this is by having features on a polypeptide that mark it as being appropriate for this co-translational translocation. There's something called a signal sequence, that is, this is the signal, put me through a membrane. And it's not a very specific sequence. It's a positive charge with a bunch of hydrophobic residues. There can be anywhere from 6 to 15 in this hydrophobic region, and typically that can form an alpha helix. That signal sequence is picked up by SRP, or signal recognition particle. And one of the interesting things about SRP, and it's called signal recognition particle now because we know this, is that there is an RNA included in the SRP as an integral and essential part of the SRP. SRP used to stand for signal recognition protein, but then we found the RNA, so now it's SRP for particle. This is the SRP, part of the SRP structure. You can see here the this is unfortunate that it's there's the RNA is part of a complex. This is the protein part, and this is the sort of the hot dog roll into which the signal sequence can pack itself. That positively charged residue in the signal sequence can interact with the negatively charged RNA. And when that happens, when the signal sequence interacts productively with the SRP, that stops translation. And nothing, it's going to, the ribosome won't go on any further beyond that point once SRP steps in. And so here you can see this early stage of it. Here's the messenger RNA that's coming along, and it started to translate. Now you can imagine that part of the growing peptide chain is buried inside the ribosome. The ribosome is a big thing. And so the earliest bits of the messenger, or the protein being made, aren't going to be exposed outside the ribosome until it gets to a certain length. And so this stage of the process, what we've seen is that the ribosome has translated enough of the message that there's now a signal peptide sticking out and exposed from the ribosome. If it interacts with SRP, that will then arrest translation at that point. And that complex then has to go somewhere for translation to continue. And this complex can interact with the endoplasmic reticulum. Endoplasmic reticulum, or ER, is a network of membranes inside the cytoplasm of eukaryotic cells. Some of its functions are like lipid synthesis, but they're also involved with protein synthesis for things that need to go somewhere else. So if it interacts productively with the endoplasmic reticulum, the SRP hydrolyzes a GTP and goes away and interacts with a translocon on the ER, and that's going to cost ATP. So there is some energy invested in this process of getting the thing to the right location. Once the ribosome and its message and the growing chain are properly anchored on the right location, the ribosome can continue to translate, and the peptide chain will continue to grow. And in many cases, that initial bit of the protein is cut off, that there is a signal peptidase that cuts off the signal peptide before the protein can be considered a mature protein. Translation continues, and that protein might be either delivered into the lumen here of the endoplasmic reticulum, or, and they're not covering this case, it might become an integral membrane protein anchored in the membrane and redistributed. What are some of the things that can happen to a protein when it's full length? It turns out that there's an entire zoo of post-translational modifications. I have a friend who is a protein chemist who at one point in his more dark moods thought that every protein is modified if you look hard enough. I don't know if he's right, but certainly many more are modified than we used to think. Some of the kinds of modifications, proteolysis. We just saw that you have to have proteolysis to take the signal peptide off. You may remember proteolysis as part of the processing of chymotrypsinogen. The chymotrypsinogen is not a mature protein until it undergoes proteolysis. Insulin has to be proteolyzed in order to make functional insulin hormone and so on. So that certainly is one very common one. A second modification is glycosylation. This is sort of a very big umbrella to cover a lot of different kinds of modifications. Most of the integral membrane proteins that face the outside have sugar on their outsides. Serum proteins generally have sugars on them. There's a different set of sugars that are characteristic of the integral membrane proteins of the ER. And people have spent entire careers looking at how proteins get their particular tree and modified sugars and so on. There's a variety of small additions that can be added, methylations. We've already seen acetylations, for example, in the case of histones. Palmitoyl addition we saw in the case of the membrane proteins that were anchored to the membrane through a palmitoyl end. It's very common to find the amino terminus acetylated, and that then removes a positive charge from that end and lets it pack in tidy ways. It's common for the C-terminus to have an amido group, which again neutralizes some of the charge there. This picture here, if I'm looking at it correctly, looks like it's an example of one of the complex glycosyl groups that gets put on some of the proteins. And this is just showing you on the insulin chain how essential it is to cut here and here to liberate a mature insulin molecule. This third part of the insulin, the C chain, is one of the most highly variable protein sequences in the animal kingdom because its only job is to be long enough and to still be able to be cut by a protease. I've said it many times before this. Whenever there's synthesis, there's degradation. There has to be some way of getting rid of things. And really the only exception to that would be DNA, where you try to have progeny, so you can always give away your DNA. But certainly protein degradation is an important aspect of it. For a long time, protein degradation got very little attention because it was seen as sort of a boring part of life. And it was only after thinking about it that people realized that degradation has to balance synthesis, or we'll all cover the earth up to the treetops. And one of the ways in which proteins can be degraded in a regular, regulated, orderly way is through a structure called a proteasome, which is a protease containing some or body. And this is a picture of the core of a proteasome. You can see there are a number of polypeptide chains here in kind of a barrel structure. And they consume ATP as part of the way in which they can do this in a specific way and not just promiscuously attack everything in the cell. One way that eukaryotes have of targeting proteins for degradation is with a small protein called ubiquitin. And ubiquitin got its name because it was found in every organism it was ever looked for in, and it's an extremely highly conserved protein. It turns out that the reason it's so ubiquitous and so conserved is that it's an essential part of the protein degradation machinery that goes through the proteasome. There are some protein features that can tend to make a protein longer-lived or shorter-lived. One such feature that's been identified is called the PEST sequence. This is not like your zip code where there's 5 digits and it's always that. It's more sort of a flavor, if you like. Hydrophilic stretch of amino acids that's at least 11 long, that includes at least one proline, one E, which is glutamate, or a D, which is aspartate, and then one S, or serine, or threonine. And then on the sides of that are lysine and arginine, which are positively charged, or a histidine. And there can't be any positively charged things inside the PEST sequence. So this is the kind of feature that protein recognition uses for these broad purposes. There's not one single thing. It's more a kind of a general flavor. And this allows the cell to have things with different lifetimes. It's not either short-lived or long-lived. It can be very short-lived, somewhat short-lived, moderately, very long-lived. And so by having a better or worse pest sequence, you can tweak the lifetime of the protein. I'd like to spend the last few minutes then talking about some work that came out of my own laboratory. And this is work that integrates a number of different features of what we've been talking about, both during the macromolecular metabolism part and also earlier in the semester. We talked a little bit just now about the co-translational insertion of membrane proteins, and that these are proteins that have to go through a membrane as they're translated. It turns out that there are some proteins in the cytoplasm that are also co-translationally assembled, and something like this, that if you look at polyribosomes, and so here you have your message with its ribosomes, these are in general, in most cells, found associated with the cytoskeleton. Polyribosomes are not generally found floating free in the cell. And so there's a little bit of cytoskeleton for them to be attached to. It has turned out to be the case that some growing peptides, so these are polypeptides that are still on the ribosome being made, for some cytoskeletal proteins, their growing peptides can attach to the cytoskeleton in a specific way. So the membrane proteins that were going through a membrane co-translationally, these are cytoskeletal proteins that are associating with the cytoskeleton co-translationally. How would you be able to know that something was a nascent chain? There are a number of different methods for doing something like that. A very general methodology for looking at things that are coming into existence is pulse chase, where you give a little bit of a label and then follow that with something that's not radioactive. So if you give a little bit of radioactive methionine and follow that with cold methionine, only the bits of protein made in that shorter period are going to be the radioactive ones. You can select your protein with an antibody, separate it by gel electrophoresis, use autoradiography to detect it, and then see what proteins you have. Several different proteins were found to do this. We talked back in the protein structure part about myosin and myosin heavy chain. Myosin heavy chain is about 200 kilodaltons, and about a third of it assembles during translation. I don't believe we talked about titin, except sort of from the sidelines. Titin is a muscle protein about 1 million molecular weight. About 80% of it assembles during translation. Vimentin is an intermediate filament protein. We talked about how they're not very soluble, and it turns out to be also assembled, but in different extents in different cells. That is, in red blood cells, it assembles after translation. In fibroblasts, about half of it during translation. So here's a picture to show you that you can actually detect nascent polypeptides. The P stands for the pulse. This is what the polypeptides look like at the end of the brief labeling period. And here is the protein at the end of an hour or so to complete translation. So almost all of these peptides in here are proteins that are still coming out of a ribosome. These are still in the process of getting born. Well, is the whole cytoskeleton made that way? No. We looked at some proteins like tubulin, which was clearly post-translational. Alpha-actinin was post-translational. Non-muscle actin was less than 10%. It was, if it's any at all, it's smaller than we could detect. Other proteins that can, we looked at, we mentioned already Vimentin. And so what we're going to do is look at the proteins that do assemble this way and see if they're common features. Remember back in the protein structure part, we talked about coiled coils, that an alpha helix would wrap around another alpha helix, and that the intermediate filament proteins are all structured like that. So Vimentin is a parallel coiled coil. Myosin has a very long parallel coiled coil in its tail. Titin is not a coiled coil, but is an extremely long, skinny protein that sits down on long, skinny structures. When we compare that to these proteins over here that don't, Tubulin is a globular dimer. So that's kind of, you could see that might be hard to assemble during translation. Alpha-actinin is anti-parallel. That is, the N-terminus of one peptide binds to the C-terminus of another. It's very difficult to see how that could do it during translation. And so if we take those features, it'd be nice if you can predict it. In science, it's always nice if you can predict things. And we decided that a candidate for this was tropomyosin. Tropomyosin is another cytoskeletal protein, associates with actin filaments, and its attractive features were it was a parallel coiled coil, like the vimentin and the myosin, and it was a long, slender protein on a long, slender structure, just like chitin. So those are two features it shared with other proteins that assembled during translation. And did it? It does in muscle. So when we looked at its assembly in muscle, we found substantial levels of co-translational assembly, but it didn't in fibroblasts. But I should say, this is not the same protein. Remember we talked about introns in messages and that different tissues could process different messages in different ways. The example that the textbook used is actually tropomyosin. And the non-muscle tropomyosins are longer than the ones in muscle cells. So it would certainly be interesting to suggest that it is the isoform that makes the difference. What difference does it make to think that parts of the cytoskeleton assemble as they're being made? Well, it might mean that where your messenger RNA is would be very important because some of the assembly was going to happen where that message was. We know that for many of the cytoskeletal proteins that they can disassemble and reassemble, so it isn't necessarily the case that all the protein will be where its message is. There might be rearrangement after synthesis. But it does suggest that message position might be important, which is something that was not supposed for a very long time. And so we looked at the message positions for some of these proteins. I mentioned already that titin is a huge protein, muscle-specific. Vimentin is present in muscle and non-muscle cells, in young muscle cells and non-muscle cells. And we looked to see if they were localized, and if they are localized, what that might mean for how they work. Does it tell us anything about their position? Now here is, am I missing something here? Let me just take a peek. Okay. I think there's a picture that should have come up. Let me see. Okay. So in this picture, this top picture is labeling protein with an antibody. The bottom picture is labeling RNA by hybridization. And it's clear that the message is not all over the place. It's in some places and not others. And so we ask, where is the message relative to the protein? And this is the fusion of those two pictures where red is the RNA and green is the protein. And at the first glance, it looks like they're in different places, that the green is not where the red is. But our antibody is to a different part of the protein than the RNA. And so as it happens, the RNA is localizing to the part of the protein that it codes for. It's not merely co-localizing with the bulk of the protein, it is co-localizing to the part of the protein. Okay, I'm sorry. Well, that's one reason I had the black and white. This arrow, okay, let me see. The doublets, the little thin doublets are green, and the somewhat thicker singlets are red. Is that all right? Certainly. I hope you've noticed that I have tried to avoid red and green for the most part in my lectures, and this is one of the few places where I was not able to. And then here's a spelling out of the names of those features. This is the AI junction and the myosin band. If we look at vimentin protein, now vimentin is again the intermediate filament protein, and the top panel here is showing the protein staining. The bottom panel is showing the RNA staining. And so the bulk of the RNA is near the cell body, and the bulk of the protein is at a distance from it. But most of the protein, of course, is hours to days away from having been synthesized. If we wanted to look during the development of the muscle cells, well, this is showing actually a section through this roughly the same time, showing the localization of the message to the cell body principally, and then the protein out in the processes. As muscles continue to mature, they rearrange their intermediate filaments until by the time they have the stripes of myosin, they also have stripes of intermediate filaments. And in this one, this is, again, a fused red-green image, so I can't explicate it quite as easily as the Titan one, but part of what comes across is the majority of the protein and the messenger RNA are in the same places. These little packets of bright spots are almost certainly transportation forms. Another laboratory has done extensive work showing how messages are transported in packets. So, muscles are not necessarily the easiest of systems to look inside. This is showing you fibroblasts, vimentin in sort of their home cells, the fibroblasts that make the bulk of the tissues. Go back, go back. Page up. There we go. Nope. Where are we? There we go. Here we go. So this is showing just the typical normal correct arrangement of vimentin in a fibroblast. And these cells are at a density where they're probably not walking anywhere. However, if you look here, you can see the messenger RNA for the vimentin, and you can see that the message is concentrated in a place, and the place where the message is concentrated is near a little structure called a centrosome, and the centrosome is the place in a cell that organizes the bulk of the microtubules. So the messenger RNA at rest is not randomly distributed. We can ask, is there any relationship between message position and cell movement? And to do that, we have to make the cells move. We can grow the cells till they're a thick layer. We can make a fake wound. That is, we can simply go in and scratch some of the cells off. The growth medium that most cells grow in is essentially wound medium. That is, it's serum. This is something that many people sort of gloss over, is most cells are being grown as if they're in a wound. And what happens? Well, if we look at the RNA, and the centrosomes, there we go. So what we find is that at the leading edge, the front edge, the RNA for the vimentin is moved to the front of the cell, whereas in the cells that are further back, it remains more randomly distributed. And the centrosomes also are found to have, in many cases, reoriented to the front. The movement of the centrosomes has been known for a while. The idea that the messenger RNA would move with the centrosomes was not. Well, maybe it's all coincidence. Maybe it just happens to be a handy place to put it. Can we interrupt or interfere with message position and interrupt or interfere with the cytoskeleton? That's one of the ways you test the functional significance of something. Now, these are cells that already have vimentin in them, so we have to be able to see the vimentin. And to do that, we add a little tag that doesn't add any functional consequences. We can send messages to different places. So the 3' untranslated region, UTR for untranslated region, you remember you have the 5' methyl cap, you have the coding sequence, and then you have a 3' bunch of stuff that ends up with a poly A tail. The 3' untranslated region between the stop codon and the poly A tail is sometimes larger than the coding sequence. And this has been a great mystery, something appalling. Why would the message bother to have all that stuff there? Well, there are reasons to suspect that maybe it contained information for positioning, so we can change its 3' UTR from vimentin to something that is normally found somewhere else, like actin, and then put that into cells. We can put it into cells and ask, when we change the position of the message, does it change shape? Does it change movement? Does it change cell division? Things like that. And so we had an experimental molecule, MIC-tagged vimentin with the beta-actin 3' untranslated region. And then the control, you can't just put in one thing. You have to control for possible variations. We had one message that was going to have the beta-actin 3' UTR, but beta-galactosidase. It has no functional consequence to the cell. We also had a MIC-tagged vimentin with its own zip code, so we could confirm that having the MIC tag on it hadn't interfered in any way. And I think this is just to go back. Oh, well, there we go. So this is an example of a fibroblast. This is not a neuron. This is not a glial cell. 3 days after expressing a mislocalized messenger RNA has done this to the cell shape. This is just a little zoo showing you how distorted and strange the cells can get with expressing. All we've done here is to express it in the wrong place. The protein is functional because the earlier picture I showed you was of a MIC-tagged vimentin off of its own zip code. All we've done is to make the protein in the wrong place. Another couple of examples of this sort of bizarre branching process that almost looks like a neuron at this point. This is showing, here's the vimentin protein that clumps out into the processes. The RNA, instead of being all inside, is out in processes it normally would not be. Well, maybe this makes no difference to how they move. So we can repeat the wounding experiment, growing a monolayer. And we can ask whether there's any difference in the tagged vimentin that's gone to the correct location. So this is the normal vimentin, if you like, or the mislocalized messenger RNA. So this is right after we made the wound. You can see that the cells don't look radically different in the context of a monolayer. But at 60 minutes, we can see that the normal vimentin is participating into that movement into the wound, whereas in the cells that had mislocalized their message, they've been left behind. They're unable to organize themselves in the correct direction and fill in the wound. So here's a functional consequence to mislocalizing the message for a cytoskeletal protein. All right, that's, again, wrong. So I will call that that. Any questions or any questions? Okay.